Good morning, everyone. This is the agenda for February 3rd and 4th. So today we're going to start with an announcement and our Black Scientist of the Day. Then for our assignments today, we have the pundit bell work, just like usual. I want to do a little bit of review and then introduce you to our assignment, which is about mistakes that are made during cell division and what can happen from that. So for the announcement today, I just want to let you know that next week is going to look the exact same as this week as far as your schedule. iStep is going to happen again next week. So Tuesday and Wednesday, you will not come to school. You will do work on your Canvas classes on your own. If you have any questions about this, let me know. I will try to make sure that I bring this up again for both my Green and Red Day classes because I'll see each of you at least once before next week. Our Black Scientist of the Day today is James E. West, born in 1931. He was, or is, an inventor, a formal Bell Laboratories engineer, and physicist at Johns Hopkins University. He is most famous for the invention of the foil electret microphone, which was patented in 1962. And the big deal about this technology is that about 90% of microphones use this technology today. So originally, he designed it to help psychologists study human hearing. They needed something small. But now it's used in cell phones, hearing aids, baby monitors, professional music equipment, and many more things. So it's everywhere. How cool. So for today, we are going to start with the punnet bell work. So as usual, we're going to go to Canvas. And to our modules. And then from here, we go all the way down to today's punnet bell work, February 3rd and 4th. That aren't the same with each other. So this bell work is going to look almost the exact same as Monday/Tuesdays. It's in the same format. I just changed the information. So for the problem, purple flowers are dominant to white flowers in pea plants. So if a heterozygous flower is crossed with a heterozygous flower, what are the possible genotypes and phenotypes of each? Okay, so first two again, do you know what the dominant and what the recessive phenotypes are? Again, just copy and paste this. These two are going to be your parent genotypes. So, for example, up here it says a heterozygous. So you're going to put a big P here and a little p here. Same over here. Then here are your genotypes that come from those parents. So those two alleles coming together, that's what you'll have in B, C, D, and E. In numbers 4 and 5, remember that genotype and phenotype are different. I want you to list out each one you see in this Punnett square, not including the parents, just the offspring, and what percent. Remember, each box is worth 25. Okay. At this point, it should be second nature. We're just continuously practicing this. Let me know if you have any questions. Next, I want to do a quick meiosis review. So we did review meiosis and mitosis last time, and you did a Venn diagram on the difference. So really quickly, other than the stuff we already know, like it happens in your sex cells, they create haploids and there's two divisions. We talked about how there are 4 genetically different cells, and that is because of crossing over. What crossing over is this process that happens between homologous chromosomes, so one that you get from mom, one that you get from dad. They're the exact same chromosome with the exact same traits, but maybe you have different alleles. And the crossing over that happens, they exchange information with each other, and this increases genetic diversity. That's what makes us so different from one another. And then these chromosomes tend to be very unique and have unique sets of genetic material. That's why we can be very similar to our parents and siblings, but that's why we're still very different because of this crossing over process. Now, what I want to talk about next is mistakes that happen during cell division. So there's this process called non-disjunction. It's not really a process. This thing that happens, it's called non-disjunction. And it's when chromosomes don't separate like they need to. So in cell division, you have your two cells, they separate, and then each cell has the same number of chromosomes. In non-disjunction, something happens and those chromosomes don't get pulled apart. So then one cell has too many and other cells don't have enough. And this can happen in so many different ways. So, what we're going to do today is learn a little bit more about what happens when non disjunction happens. So, to start on that assignment, we'll go to Canvas, go all the way down to modules, scroll all the way down, and go to mistakes in cell division. I want to fix that. But again, it just reminds you what non-disjunction is, what it looks like. So 3 steps. One step, you're going to pick a chromosomal disorder. So these are different disorders that come up from non-disjunction. So you're going to just pick one, whatever sounds most interesting to you. Then you're going to do some research. What is the chromosomal disorder you researched? Why is it named that? What is wrong with the chromosomes? What are signs and symptoms? How many people are affected by it? And what sources did you use? You have to use at least 2. And then make sure that you have at least 2 pictures. Then you're going to put all that stuff into either Google Slides or a very organized Google document and turn it in. If you have any questions about that, let me know. Just enjoy learning about what happens in these instances where mistakes happen.